All right, so we'll pick up again here. So I think we have quite a lot of time in this afternoon session. I'm hoping we don't run the full length, even though there are 3 more modules. So the next 2 modules will be similar in sort of content to the previous one, in that we will look at sort of systematically building up models, discussing inference, things like that. The main difference will be to look at models with slightly different structures that are sort of important in cancer biology or computational cancer biology. I will move a bit faster through these, hopefully, because most of the process should be repetitive this time. And then in the last module, when we come to it, it'll be sort of just some miscellaneous material, really about better ways to do inference mostly, things that are a little bit more advanced than we've talked about thus far. So for this section, we'll turn to copy number variation. And my pointer working. So hopefully this is review for everyone, but copy number variants are another class of genomic aberrations, of large-scale changes now within the genome, duplication, deletion of regions. So in the same way as SNVs, they can be driver mutations, and we can also use them as markers of clonal populations. So many of the same things that we like to do with SNVs in terms of studying cancer evolution, we can do with copy number variants. From a modeling perspective, the major difference between CNVs and SNVs is spatial correlation. And the point being, because CNVs are large, if we look at 2 data points in the genome, they will tend to be affected by the same copy number variants, right? So there's this idea that looking at 2 points, there's some relationship. Whereas with SNVs, the point mutation process, in some sense, tends to render them independent up to some approximation. So, sort of working through our typical thought process, before we come to a question, I'm just going to look at something a little more basic and think about how do we represent data when we look at copy number variants. Here I'm going to assume high throughput sequence data. We'll talk about bulk or single cell, depending. The main point here, in much the same way that we summarize the aligned read data for SNVs in terms of integers, counts of reference variants, we need to do something like that for copy number variant analysis in order to start to build models, right? Again, coming back to this idea that it's a little bit hard to model just reads aligned on a genome. So the simplest approach is to do essentially the same thing we do for SNVs and look at positional allelic count information. We could do this across the entire genome, right? All 3 billion positions. The count information for what we'll call the variant will be pretty boring at most positions because we won't have SNVs or SNPs, right? So most positions will just report 1 nucleotide. So really most of the information at those positions is in terms of the depth of coverage, not really the proportion of the variant reads. One issue with looking at every position in the entire genome is that your data set quickly gets large. 3 billion positions is a fairly challenging number to deal with, even if one is doing sort of the simplest thing like map inference. So one solution is to restrict your positions, right? And the most common thing to do is to only look at heterozygous SNPs within the genome. One reason is this drastically reduces the number of positions we have to consider, right? It takes us from the order of billions onto the order of millions typically. You may ask, how do you know what the heterozygous SNPs are? There are a few options. The best one is to look at the matched normal data, and then it's very trivial to identify heterozygous SNPs if you have decent coverage. You can also just take panel, you can also look at known positions from things like dbSNP. So once we've restricted to heterozygous SNPs, then it does start to make sense to look at the reference and variant counts, right? Because we will actually have variants at those positions by definition. The nice thing about working this way is that we can actually look at allele-specific copy number because we now have information about the prevalence of both alleles. One issue with this representation is it's not terrifically useful for low coverage data. So for bulk sequencing, it tends to be okay. If you move into single cell genomes where you have, say, 0.1x typically, then you're very unlikely to have even one read overlapping a position, but definitely not 2 to 10 that you would need to pick up both alleles. So you're not really going to get any information about the alleles. So this brings us to another way to represent the data, and this is to divide the genome into bins. You can do this with equal length bins, or you can make them variable. What's typically done is equal length bins on the order of thousands to 100,000 base pairs, right? Then what you do is you look within this bin, the segment of the genome, and you count all the reads that have mapped there, right? So we essentially just get one number, which is total depth of coverage. Now, assuming that copy number is roughly proportional to read depth coverage, this is a very useful way to get total copy number information. The nice thing about this strategy is that we can use it even when we have low depth data. Because the bins are so large, even if we have 0.1x, we'll typically have many reads mapping within, say, a 100,000 base pair region. So this is a contrast if we just look at a single point mutation, or a single point in the genome. The other big advantage is that it's computationally much cheaper. If our bins are big, we won't have many of them, right? So if we have, say, 100 kb bins, we'll typically only have about 10,000 bins across the entire genome. The key deficiency of this representation is we can't get at allele-specific copy number because all we're recording is total depth of coverage. We're not really recording any information about parental alleles. So the final representation is somewhat of a trade-off between these two, and that's to look at what are called haplotype blocks. So a haplotype block in this setup just means a region of the genome where we can phase the heterozygous SNPs onto parental alleles, right? So that means that we will know exactly which allele maps to the paternal and which maps to the maternal. Though we don't actually know those labels, but we can say all of these heterozygous SNPs are on one chromosome, all of these are on another chromosome, right? Does that make sense? So, and this is relatively easy to do using existing software if you have the matched normal data. Once you have these blocks, then you can record 3 pieces of information. The total number of reads within the block, just like the bin read count data. But then you can also record the number of reads that support the first haplotype and the number of reads that support the second haplotype. So this becomes allele-specific information, just like in the simplest allele count representation. This is usually preferable if you have bulk sequence data to using the allele counts and also to using the binned read counts. It's not as useful in low coverage data because haplotype blocks don't tend to be incredibly long. The phasing software basically just has an issue. The phasing software itself is a probabilistic model that looks at the normal typically, and there's only sort of a limited stretch that it can resolve. And this is basically due to issues like recombination and things. So these blocks might be between 100 to 10,000 base pairs, but usually not 100,000 base pairs like we would use it with binned reads for low coverage data. So that's sort of the data representations we could use. So the key thing in S&Bs is we want spatial correlation, as I mentioned, and the basic intuition, no matter what your data representation, if you look at two adjacent points in the chromosome, they are more likely to have the same copy number as a different one, right? Because assuming that the chromosome, the copy number variant is large, say larger than even our bin sizes, and that's typically true, they're on the order of megabases, not kilobases, then you would expect multiple data points to be at the same copy number. So in terms of modeling CNV data and trying to infer the actual copy number, there's two approaches that are commonly employed. The first one is to segment the data in advance, and this basically means look for sequences of data points that we believe to be in the same state, i.e. the same copy number. When we do the segmentation in advance, we don't necessarily try to assign the copy number at that point. We do that in a second step, sort of using some sort of modeling, right? The other option is to both segment the data and infer the copy number and anything else you're interested in all at once, right? So strategy one tends to be easier because you can use sort of simple tools like circular binary segmentation or modifications of that to quickly identify blocks of data that are in the same state. Once you have those blocks, they are effectively independent, so then you can actually basically model them in much the same way that we did with the S&V data, right? You've eliminated the spatial correlation. The downside of this approach is that everything you learn in the next step, where you infer copy number, tumor content, if you do clonal structure, whatever, that doesn't affect how you do the segmentation. You can never go back and improve the segmentation, right? So if you take the second approach, as you learn about tumor content, that's going to influence how you segment the data, right? There's obviously pros and cons of each. Here we're going to look at the second strategy. It maps a little bit more nicely onto the probabilistic modeling framework, right? And just turn to you guys, has anyone worked at all with copy number data? A little bit, and are you familiar with some of the existing approaches?
I mean, the names like ASCAT, I think you use.
Do you know which category ASCAT falls into on this? I see. Yeah, so ASCAT is one of the more famous methods for bulk sequencing data, and it's in category 1. Related algorithm for clonal population structure, Battenberg, is also in that category. Approaches out of the Shaw lab, like Titan and Remixed, will be in the second category. And in the end, which is better is kind of an open question. I think they all make some trade-offs. So in order to model spatial correlation, we need sort of specialized approaches. So hidden Markov models are by far the most popular method for modeling spatial correlation. Essentially, anyone who has taken a course in bioinformatics sees an HMM. They are ubiquitous in the field. There are other ways to model spatial correlation, and HMMs actually fall into a much bigger class of things. Kalman filtering is another thing in that class, and the bigger class is something called state space models. The key thing about HMMs is that they assume data points, they assume that the data point you're at only depends on the previous data point, or actually the hidden state of that data point only depends on the previous one. And this is the Markov assumption in hidden Markov models. This is really helpful because we'll see later that it can lead to very efficient algorithms for fitting. The biggest downside of hidden Markov models is that very assumption means that we have a limited ability to model state duration. Informally, this basically means that the model naturally wants to switch states after some period of time, which is geometrically distributed. In practice, what you may see when you fit an HMM is you'll have a long stretch where it's in one state, it will quickly flip out of that state and then back to it. And it's not because the data necessarily supports that, it's just an implicit thing in the model, right? So latent variable model, HMMs fall into this class of latent variable or hidden variable models, where the hidden variables have this dependency structure, that first order Markov. So the key here is the latent variables take a value in a discrete space, just like mixture models. So it's essentially a thing that points and says what state. For us, what copy number state are you in? And another way to see them is as dynamic mixture models. That's often how they're described. And it says now your chance of being in a different cluster or mixture component is going to depend on what the guy in front of you picked, right? And basically in our case, we use that to say, well, then you should choose to be in the same cluster as the previous data point. That's the main point. So the general again, I'm going to give a very abstract definition of HMMs first, and then we can specialize it to a model. But all HMMs have sort of the setup. We have the vector pi. This is the initial state vector. And so this is the set of probabilities that say, what is the first state in your sequence, right? So the first state has this special treatment in the setup. After that, we then have the transition matrix. And this says, given the previous state was I, what is the probability that the next state is j, right? And once we have those two things, we can compute the probabilities of the states along an entire sequence. Just like the mixture model case, we then have this arbitrary distribution F, which is in HMM speak, the emission distribution. And the key thing is that the hidden states that we have, just like mixture models, act to pick which parameter goes into F and generate the data. As we talk through this, we're going to assume that the number of states, the number of components in our HMM is known and fixed, equivalent to assuming that we know the number of clusters is fixed in mixture models, right? You can relax this assumption in the same way as we did using non-parametric priors. Because of the spatial correlation, this is much more computationally demanding for HMMs, so it's not something we typically do. As we'll see later, it's potentially not as contentious because K here is typically the number of copy number states we have. So we can sort of fix it at some maximum we think is reasonable. So the general sort of form of a Bayesian HMM then looks like this. We sample pi from a Dirichlet. So remember, that's the initial state distribution, and the Dirichlet is that distribution that gives you a vector that sums to 1. We then sample the rows of the transition matrix from a Dirichlet also. We then sample the initial state, Z1, from a categorical distribution, then sample Zn given Zn minus 1 and the transition from a categorical, where we pick out rows Zn minus 1 from the transition matrix, right? Then, just like the mixture, we have our cluster parameters, our hidden state parameters that we simulate from an arbitrary distribution, and our data, which we simulate from another arbitrary distribution where we index it. So the important thing, and the only real difference between mixture models is this sort of thing, is this step here, where we're looking at the previous data point and then we're deciding what is the probability we pick a cluster, right? The most important thing, or probably the most important reason that HMMs are popular, is they're the simplest model that has spatial correlation, and inference is reasonably easy, right? So the key thing here is the existence of the forward-backwards algorithm, which is just a dynamical programming algorithm that lets us essentially marginalize the hidden states. And so this is useful when we want to do map estimation via the EM algorithm. And you can also make use of forward-backwards and MC-MC sampling, right? There's also a related algorithm, the Viterbi, that lets you compute the maximum path through your state spaces given the parameters. Here we'll discuss EM to perform map estimation. So rather than MC-MC in this module, we'll focus on map. The main reason is it's the more common approach, mainly because MC-MC can become computationally demanding, especially if you have fairly long sequences. And the basic idea is we use EM to find the values of pi and A, the transition in the initial state. And then once we know those, we can then use the Viterbi algorithm to compute the most probable path through the sequence. I won't go into details on how to derive those, but the basic recursion looks like this. This is the forward recursion for alpha. Essentially what it says is the probability, alpha here is the marginal probability, I think, of seeing state K, right? And beta is sort of the reverse thing. The importance of those quantities comes from the following. And it's really these two things. It allows us to compute the expected value of our hidden variable n, and also the joint value of the hidden variable n minus 1 and n equals l. Why that's important? So if we do map inference, we end up actually having closed form updates for pi and A, right? So we can compute the map estimators. This follows because by construction we have conjugacy in the model between the categorical and the Dirichlet, and you end up with these closed form updates. They depend on the expectation, on those two expectations that we defined previously. You will also typically have other parameters in the model. In the cancer context, these will be things like tumor content. Those won't lead to closed form updates, so you'll have to do some form of maximization. This typically means you have to compute a gradient at some point, at least to do it efficiently. And your gradient's gonna, again, is gonna take a form, take something like this form, where again, you need to know these expectations, and then you just weight the gradient of your target. All right, sort of the review of spatial models. Now let's just talk a little bit about CNV modeling. So we're going to start with a kind of a boring problem, but I think it's illustrative of the basic process. So what we'd like to do is infer the copy number profile from bulk, and we're going to assume that we have, well, actually it doesn't have to be from bulk, but we're going to assume that there's no clonal population structure and no normal contamination. The data that we will take is binned total read counts, and what we'd like to infer is total copy number. So yes, if this is bulk, this is not an interesting model because we don't have clonal population structure, we don't have even normal contamination. However, it's actually a perfect model for single cell data where we don't have those problems. And because we're thinking about bin read count data, it directly applies, right? We'll have a bit of notation here. One layer of complexity that gets added when we actually consider genomic data is we have multiple sequences. So sort of the equations I gave were for HMMs when we only had one sequence. We have multiple sequences in the genome because we have multiple chromosomes, right? Each one is a separate sequence. It doesn't really make sense to treat the genome as one giant sequence, right? Because knowing what's at the end of chromosome 1 tells you nothing about what's at the start of chromosome 2. Does that make sense? All right, so now little m is going to be our index on chromosomes. M is going to be the number of chromosomes. We're going to have bin data point indexes, number of data points per chromosome, which will be variable because chromosomes have different lengths. We will have the number of reads that we observe in the bin, and we will have this hidden state, right? So the model that we'll consider to start with is basically the generic model, where we have the basic HMM construct up here. But because we want to look at copy number, and we're going to look at total copy number, we don't necessarily want to sample the parameters for each state completely independently. In some sense, if we make a very simple assumption, which is that the read depth is proportional to the total copy number, right? So if I have one copy, or if I have two copies, then I should have twice as many reads as one copy, and if I have three, and so forth, right? If we just sample the parameters for each state completely independently, we won't catch that. So to get around that, we introduce this parameter R, which is, in this case, interpreted as the haploid read depth, which would be if you had one copy in a bin, what would be the expected read depth, right? The parameter then for a copy number state, given R, is just multiplied by that copy number state, right? Given all this information, now it just looks like a mixture model where we draw our data from a Poisson. Why are we using a Poisson here? Because our data takes values from 0 up to infinity, integer values from 0 to infinity. We'll come back and we'll see why that's maybe a bad assumption later. That's the basic model. Oh, sorry, C is the copy number of the state. So here I'm assuming, yeah, maybe I should say this a little more clearly. It is both, but we're tying the hidden state to something interpretable. We're saying that the hidden state is the copy number, right? Or we're doing a mapping onto it. So we're saying, because Zn is data point specific, C is generic, right? What we could write is Zn equals C, right? And then we could index the last equation by C. Zn is going to take values in C. It's going to take values from 1 to big C, our maximum copy number, right? Does that make sense? Does that make sense to you guys, or not really? Okay. Yeah. So, so ask more questions if these things are. I'm going quick because I feel like we did a lot in the previous modules. So, but there's also been a lot to digest. So let's go a bit slow. So I'm using, so this is meant to be a vector of all of these. Yes. I don't know if that's coming out. But what we're saying is that we're going to use R to generate this vector of all of our thetas, right? So we're going to have a vector that looks like this. Up to some maximum number CR, right? Now, that each of these represents the parameter of one of the components, or one of the, let's say one, let's keep with components. So then if I say that Zn is equal to 2, that means data point n is going to use the second entry, which is here, right? 2R. Is this clear? You can, of course, just assign you could sample this vector sort of just like we did in the mixture models, where each of these is a separate draw, but then we aren't able to include this assumption about copy number affecting the read depth, right? And we lose some interpretability. The nice thing with this model is that when we say we have copy number state 2, we're enforcing that implicitly, right? Otherwise, we would have no labels on the actual states. So the simple assumption that copy number is a function, that read depth is only a function of copy number and haploid read depth is not good. So people who have looked at actual genomic data and done CNV analysis can attest to this. The major issue is GC content, right? And this has a massive impact on the read depth. So if you were to go back to module 1, there was this question about what does the binned read count of a diploid cell line look like? Because everything has the same copy number, you would expect to roughly see the same read count, right? But what you will actually see is a wavy distribution, which is just a result of GC bias, right? Somehow you have to deal with GC bias. The most common strategy I know of is to do some form of a pre-processing step where you essentially do a low S regression to remove it. If we do that in our case, then we have to abandon this whole Poisson assumption because our data will all of a sudden be non-integer because we're just sort of regressing it out. So that's fine. It's easy enough to change the model and use normals or anything else. I'm not always in favor of those approaches where we use a pre-processing step to remove something from the model. If we have the covariate information, if we know the GC content of the bin, we can often just build that directly into the model, right? So the problem I have with preprocessing is that some sense you then remove the interpretability of the data and your ability to model it. And that's why you tend to fall back on saying, well, then my data is just normally distributed because I have no real intuition on what it is. Now it's just a number. Whereas if I give you bin read counts, you have some idea that, okay, this is actually a real read count that I got from some process, right? And it's a little easier to think through the model.
The way this all equation between like the speed or computational speed and like, because I assume that also costs you something in terms of just compute time when you have additional variables. Like, is that a trade-off?
Yeah, that's a trade-off. Yeah, so it's always, the more you break your process down into steps, like pre-processing, the faster things get because each step tends to be much simpler than putting them all together. So you would, and there is it's not sort of a linear trade-off, right? Because if you break something into 2 steps, it's not true that doing it in 1 step is twice as hard. And this is sort of a very informal, but it's often sort of exponentially harder, right? Because you have all these extra things to do. So yes, you're right. There's a compute advantage to doing pre-processing. This is exactly, oh, go ahead.
I just want to know, what is the impact that GC control has on read coverage? So it coverage?
Yeah, no, I may get this, does anyone, I'm gonna I may get it backwards, but I think GC rich regions tend to be overrepresented. Essentially, you just get more reads. And I think it's a virtue of PCR, but it may also be in the sequencing platform. But this is sort of a pretty well-known artifact. There's another one that's less important that I'm not mentioning here, which is also mappability. And that's essentially how easy it is to map a read to a part of the genome. So if you have repetitive regions, you don't tend to have as many reads that, depending on your liner, that map there. In practice, that doesn't seem to be as important a correction as GC content, but typically you would want to do both. And any other, I'm just using this as an example of sort of a confounder that we need to consider, but there are other ones, right? If you don't want to do pre-processing, what you can do is instead add the covariate in as your observed data. So here I'm using this hypothetical GNM as our GC content measured, whatever that means, right? It's some number. The most naive thing you can do is just plug G right into here and rescale your parameter, right? If you have this type of covariate data, you don't usually want to do that because there's no guarantee that it's on sort of the correct scale, that it's accurate, things like that. So the normal idea is to introduce another layer in the model where you have some distribution H that depends on your covariate. Out of this, you then sample a new value, and that's what you use to do the scaling. Here I've just done a multiplicative scaling. There's sort of anything else you can think of that works too. It's just an empirical question, really. Now, the other issue that I haven't touched on, the other issue with this model that makes it very brittle is overdispersion. Same issue as S&B data, we have way more variance in the observed data than a simple Poisson can model. It's the same solution. You look, instead of using a Poisson, you look to the overdispersed version of a Poisson, which is the negative binomial. You still keep the same mean, but now you have a variance parameter. So just want to emphasize, the details aren't important for the Poisson, but the idea here is you find something that has more variance, you keep the mean the same, and then you have an extra free parameter to tune the variance. So in actual tumor data, we don't have homogeneity. Well, we've already seen this. They aren't homogeneous. Probably the biggest thing you want to deal with immediately is tumor content, right? And it turns out in our hierarchical modeling framework, it's very easy to incorporate that. The main thing that we do is we introduce the tumor content Again, with no prior information, we're going to use a uniform. You may want to constrain that a little bit if you, say, had information from pathology estimates. But then given that, what we do is we change the state parameter. So now theta C becomes this weighted mixture of the copy number of the cancer cells. So we have C times the tumor content, that's the second term. And then the first term is from the normal cells. It's exactly the same thing we did in the mutational genotype, right? And then all of that is scaled by this haploid read depth, okay? And that's the modification. So now when we do map estimation, we're going to pick up an extra parameter t, and you're going to have to do that gradient descent thing I mentioned at the start, but otherwise this isn't really conceptually any harder. So the killer problem in copy number variation analysis is ploidy. So this simple model I've defined is unidentifiable. And what that means in this case is I can play this game where I double the copy number I predict for every single position, but I half the value of R. Then all of the likelihood of the data stays identical because R and the copy number appear as a product. So that effect cancels out. So basically, any time you have two parameters that appear together multiplicatively, they're probably unidentifiable or in ratios, right? So ploidy within this field loosely refers to the average copy number of a sample or a cell in the single cell case, right? And we see here that we can play this game of changing the ploidy, which is the copy number of sort of all the bins basically, and also changing this value of R. This may seem like it's just a deficiency of this sort of dumb model that we've been working on so far. It's actually just a generic problem. As far as I know, every single tool, whether it's single cell or bulk, has this issue. It's intrinsically unidentifiable. Somebody may correct me on that, but I think that at least all of the ones I know about, that's true. There is no automated way to solve this problem. So in practice, people manually curate the data. That means they go through it. I'll show you a plot next of one example. Pick a solution. It's very arbitrary. If you do rely on automated picking, you will find that this is one of the reasons that many tools give different predictions on the data sets, is ploidy. And it's also going to have a huge impact in how you predict things like genome doubling. So if you want to say this cancer type is massively genome double, you may want to go back and look at how they did the analysis. Is it because the guy doing manual curation called everything genome doubled or not, right? If you're looking for a really hard, thankless research problem, try to solve that one. Now, this is an illustration of ploidy. This is actually from ASCAT. This is how they handle it. And on the left here, we're showing what they call sunset plots in ASCAT. It's essentially a likelihood or a posterior surface. There's two dimensions. One is the tumor content. They call it a aberrant cell fraction, but it's tumor content, same thing. And the horizontal axis is the ploidy. One thing that's counterintuitive with these plots is blue means higher values, or sort of the optima. So the dark blue is where the thing you would want to pick is, and red is the inverse. So what you see right away from this is there's two pretty similar looking optima here, and they're basically at double the ploidy, right? This is exactly a symptom of that game. And probably if we took ploidy out further, we'd see more and more of these, right?
It's probably plus one and 2m plus one. It's not just doubling and like shift.
This one?
I mean, in general, it's not just because numbers will just go up and
Not quite because of odd evenness. It depends on the way you parameterize things a little bit. So in the model I showed, yes, you're right, you can go up sort of fractionally. There are other models where it's a little bit different. But yeah, that is right. So the thing is, if we picked this solution versus this one, essentially all we would do is just double each number on this axis, right? So it's something to be aware of, and there's just not a lot you can do about it. There's some ideas in the notes on ways to try to address this. The main way I see is to try to insert some side information, just like we did back in the S&V case where we appealed to copy number information to get our genotypes. We have to think, is there some other source of information we have that might tell us about ploidy? So if you have bulk data, maybe you've done flow sorting or something that gives you some additional information. Maybe if you've done single cell sequencing, you image the cell before you sequence it. Within the actual modeling framework, it's a bit harder. It's not clear that there's any extra information in the data that's going to help us, right? So one other modification we can make is to go to allele-specific copy number instead of total. So if we want to do something like PyClone, it's really nice to have this allele-specific information where we can look at major, minor. Here we're going to get away from bin read count data, and we're going to think about haplotype data, haplotype block data, right? And we prefer that to allele-specific counts because we can pool the heterozygous, the data from heterozygous SNPs in the entire block. And by pooling, that tends to give us a better estimate of the allele frequency of the haplotype. In some ways, it's almost like we've already run a little HMM to get these blocks and phase everything, and then we're using that information. So here we're going to define XA and XB to be the number of reads supporting haplotype A and B. XT is the total number of reads in the block. And now, because our blocks are of variable length, we also need something that actually tells us how long the block is, which is XL. And now our hidden state, instead of just being one integer, C, it's going to be a pair, CA and CB, which is going to be the total copy number of the A and B allele, right? So that's the main modification. The model itself actually is, again, very similar. Now we sum the copy number of A and B to get total copy. We scale by the length of the block. And then we have our normal plus. Go ahead.
So that's like a practical question. So this haplotype block is identified by SNPs, right? So
Yep.
So I define the haplotype as a sequence of SNPs that it has. But then a read maps to a place in this block where there is no SNP.
That's why we catch the total counts as well in the bin. These are the total number of reads and blocks, whether or not they map to any SNP. So, that's why we model those as Poisson. Then what we do is we model the counts for haplotype B as being binomial, where the binomial parameter depends on the number of copies of B over the total. That's the modification. Again, this is sort of the same rationale we used to come up with the mutational genotype stuff in the previous section. Right?
A plus B doesn't need to be equal to T.
Sorry, that should be B, not D. I'm apologizing.
Yeah, okay,
This does not need to be T. No, those are different numbers, totally different numbers. Yeah, I know it seems counterintuitive, but that's the main modification one needs when they do haplotype block thing. Good. All right, with that, we go on coffee break.